Hi, this is Sonia Lopez. This is my results video. Today's March 19th, 2021. This is lab number 3, Bacterial Enumerations, Dilutions, and Plate Counts. So we put these petri dishes to incubate 2 days ago. And so this was the one where we diluted the yeast from one tube to the next tube. We started with 10 minus 1 all the way through 10 minus 6. And this was for diluting the solution so that it's easier to count the CFUs. The normal is 30 to 300. In the process of doing the experiment, it helped me understand why we dilute certain specimens so that they're easier to count at the end of the incubation period. And we had a little problem with mine. I put it to incubate and we were supposed to flip it over. We're supposed to put the 4 drops of the diluted yeast into each dish and let the agar soak the yeast 12 hours, flip over the plates. But I think I may have left them out a little too long without putting them back in the cabinet, so they didn't grow the way that I expected. However, I'm gonna try incubating it again for another 24 hours to see if we see increased CFUs in the final petri dish, which was 10.6. It's almost there. I believe I counted 26 or 28 from what I completed in my lab. But yeah, so again, just remembering the incubation period, the process. Try to avoid errors as much as possible because it could affect the end result of this lab. But this was also a good learning experience because I know how certain bacteria are diluted so that they're easier to count over the incubation period that's required. Usually it's like 48 hours from a lot of these labs that we're doing, it seems like. So just being careful with that. Again, we got some colonies that formed in each tray. Some of them had very few. One had almost nothing. I couldn't see anything. Let me see. One had like just one little speck in there in the middle. Oh, sorry, that was 2. This is actually 1. Here. There's one big growth in there. There's 1, 2, 3. This is the one that hardly has anything. This one hardly has anything. 4. There's a little growth in there. 5. There's a big growth there by itself. And 6 did have the most, but again, we're going to incubate it for 24 hours and see if we obtain our 30 to 300 CFUs. Thank you.